Okay folks, so this is the podcast version of the induced pluripotent stem cell lecture from the medical genetics module, which is a level 3 module, the biomedical science course at the University of Bradford. So what we're going to be talking about in the lecture today is a brief introduction to cell-based therapy. We've touched on the concept of induced pluripotent stem cells last year in molecular genetics, but today we're going to be talking in a lot more detail about how we actually produce these induced pluripotent stem cells, and importantly how we hope they're going to benefit biomedical science. They are of a real genuine benefit at the moment, but there's great potential for the future with regards to this technology. We'll discuss nuclear reprogramming, which is the process used to generate induced pluripotent stem cells. And then we'll look in detail at what these benefits actually are. There are three major perceived benefits to iPS cells. As we continue to learn more about the technology, it may be that there's further benefits than this, but at the moment, when we think about how we can use iPS cells in a medical setting, there are three major ways in which we foresee them contributing, and we'll discuss what they are, and we'll finish with a summary of what we've talked about. So the learning outcomes from this lecture then is that by the end of it, and with some additional reading from the core textbooks and suggested reading in the module handbook, you should be able to discuss the basic concept of what cell-based therapy is, understand why in the past this sort of work has been somewhat controversial. You certainly should have a solid idea of what nuclear reprogramming is as a technique, what the concept of the technique is and how you actually achieve it. You should have a detailed knowledge of the sorts of proteins and the sorts of genes which are being introduced and expressed in order to induce nuclear reprogramming of cells to a pluripotent state. And you need to be able to at length discuss the potential benefits that these induced pluripotent stem cells offer biomedical science, and some of the roadblocks and issues that scientists have faced when trying to, if you like, bring this technology to the bedside, to the bench side, from the bench side to the bedside of the clinic, so it can benefit patients and be of use in medicine. So then, cell-based therapy, what's the idea? Well, the basic idea, I guess, of cell-based therapy, or if you like, regenerative medicine, which is kind of the holy grail of this entire area of biomedical research, is to be able to replace damaged or worn out or injured or diseased cells with healthy cells. So in essence, it's quite similar to the goals of gene therapy, where the overall aim is to either replace or augment, depending on the type of genetic disease we're talking about, a defective gene. But we've gone up a level, if you like, now. We're not just talking about replacing a single gene that isn't working, we're talking about replacing an entire cell type that isn't working. So for example, in the case of spinal cord damage, we're talking about replacing actual neurons in the spinal column. So it's a far more complex thing that we're trying to achieve. Yeah. So how on earth do you go about doing something that complex? Well, it relies upon the use of these very special types of cells called pluripotent stem cells. Now, I'm conscious that you may have encountered these in other modules already, but I'm going to give you I will still give you a basic introduction as to what these are. If you've covered some of this material elsewhere, that's good. It'll help you understand this. If not, don't worry. I'm going to give you all the information you need to be able to understand what we're talking about with regards to these different cell types, etc. So what is a pluripotent stem cell then? Well, in essence, a definition of a pluripotent stem cell is a stem cell that has the potential to become any other cell type in the entire body. Yeah. So you do get other types of stem cells, such as hemopoietic stem cells, which you find in your bone marrow, and these stem cells have the potential to become any type of blood cell, in effect, in the body. But they can't become a liver cell. Yeah? You have stem cells in your skin, which have got potential to become all the different types of cells that we find in our skin, but again, these cells can't go off and become a neuron. The beauty of pluripotent stem cells is that they have the potential to become anything else in the body. So does this cell therapy idea work then? Can we really use these stem cells to replace damaged or diseased tissue? Well, sort of. Yeah. So that's kind of an answer you find whenever you talk about a new area of treatment or research. It sort of works. There's lots of studies from mouse models now with which show really promising data. There are very, very few, a handful of actual human clinical trials which are currently underway looking at this kind of technology and its potential benefits in the clinic, and we'll talk about what some of those are towards the end of the lecture. So stem cells then. So as I've said, a stem cell is a cell that's got the ability to differentiate into any other cell type. So it's a very specialised cell type. It's able to self-renew. That's another important property that stem cells have. But it's this real property of differentiation into different cell types that we're interested in with regards to the cell-based therapy content we're going to talk about today. So it's the ability to differentiate into different cell types which is key. And the beauty of these pluripotent stem cells, as I've already said, is that they have the potential to differentiate into any cell type in the body. Brain, neuronal, lung, liver, anything. So this sort of ability to differentiate is nicely demonstrated by a specific type of benign tumour called a teratoma. So this is an ovarian teratoma here. Teratomas are tumours that are derived from stem cells. So what happens is during progression of the tumour, during growth of the tumour and tumour development, the cells within the tumour mass tend to differentiate into different cell types. So it's not just a case that these cells are undergoing uncontrolled cell division and becoming cancerous, they're also kind of undergoing uncontrolled cell differentiation to become lots of different cell types. This tumour here has got all sorts of different tissue types and cell types in it, including even teeth if you look at this here. So you know, this is obviously a very bizarre and freaky looking cancer here, and I'm not sure what this is. It doesn't look very pleasant, but it's again a different cell type, and it's this heterogeneous mix of tissues which is kind of indicative of this type of tumour, this teratoma. And teratomas are important in cell-based therapy, as we'll discuss later on in the lecture, but they also offer a nice example of how this differentiation of different cell types occurs in stem cells. So as I mentioned again earlier, there are other stem cells which also exist in addition to the pluripotent stem cells. However, these tend to have more limited differentiation options. So for example, hemopoietic stem cells differentiate into blood cell types. Skin stem cells will differentiate into different cells which are found in the skin, so on and so forth. Neuronal stem cells, different types of neuronal cell and tissue. It's only pluripotent stem cells which can differentiate into anything. And another key term for you to get your head around is this term of plasticity. And in terms of differentiation, what plasticity refers to is the differentiation potential of a cell. So in other words, a pluripotent stem cell would be considered to have very high levels of plasticity, whereas a hemopoietic stem cell would have a lower level of plasticity because it's just not able to differentiate into as many different things as the pluripotent version. Okay, so I mentioned earlier that there were some problems, some ethical issues around the use of stem cells for research, and really all of that is a consequence of how we at least used to have to isolate these stem cells, or where in fact these stem cells, particularly the pluripotent stem cells, were derived from. So up until very recently, the only way that we could actually get hold of human pluripotent stem cells was to isolate them from embryonic tissues. The process of isolation would also destroy the embryo. So as you can imagine, this raised major ethical implications with regards to this sort of research, and for good reason. People weren't happy about the idea that embryos were being destroyed or killed just because scientists wished to isolate these embryonic stem cells, pluripotent stem cells, and do research on them. And consequently, any kind of treatment which was being proposed using these sorts of pluripotent stem cells was also frowned upon. So there was a lot of opposition in the States about stem cell research for many years with regards to the treatment of spinal injuries, etc. And the entire field really was very controversial, illegal in some countries, and generally one that a lot of people wouldn't go near, wouldn't touch with a barge pole. There were alternatives to these pluripotent stem cells. It's possible to isolate stem cells from adults, these so-called tissue stem cells. I mentioned hemopoietic stem cells earlier on as one type. However, these stem cells have a lot less plasticity than the pluripotent stem cells, and therefore far less useful. They're just not able to differentiate into as many different cell types. So whilst they do have the specific uses, these adult or tissue stem cells are just not as useful as a therapeutic tool as these pluripotent stem cells. So how does cell therapy work then? What's the basic concepts behind it? Well, in its most basic sense, the idea is that you simply introduce stem cells, healthy stem cells, into a patient, provide the stem cells with the necessary signals so that they differentiate into the tissue type you're interested in. And the idea then is that these healthy stem cells will integrate with and replace, if you like, the diseased tissue with a healthy version of the required cell type. So it's a very, in essence, that's a very simple thing. It's like I mentioned earlier on, rather than replacing the gene that's defective, we're just replacing the cell type that's defective. So that's one of the ways in which potentially we can use pluripotent stem cells to treat disease. Now, I'm going to talk in more detail about some of the obstacles which exist in these treatments later on, but just to mention one thing now, so you're thinking about it. One example of a problem with regards to this idea that you can simply introduce these stem cells into a patient and expect that they're going to work, in inverted commas, is that, okay, for simple tissues that might be the case. You might have a situation where there's damage to the liver, which is a solid tissue organ that's relatively simplistic in structure, and by introducing pluripotent stem cells which have the potential to differentiate into healthy liver cells, you're able to provide the body with a source of healthy liver cells that can then repair the damaged organ. However, it's not difficult to conceive how this isn't really going to work for other solid tissue organs, particularly ones with very complex structure, the pancreas and obviously the brain, where neural networks are incredibly complex, and it would be very naive of us to think that we could simply inject pluripotent stem cells with the potential of becoming brain neurons and expect that these new brain neurons, albeit healthy brain neurons, will be able to integrate in such a way that they will completely rectify the disease or the damage that was already being done, or even integrate in such a way that they're going to work properly. So that's one of the big issues that tissue regeneration cell-based therapy faces at the moment, this question of how we actually integrate these cells into existing organs and tissues. There's a second option, which is a mobilization of innate stem cells. So, as I've alluded to earlier on when I spoke about adult or tissue stem cells, it's not just the case that we have these pluripotent stem cells as embryos. We also have pooled reservoirs of stem cells, innate stem cells within our bodies in adulthood as well. A good example of this would be the stem cells which give rise to fibroblasts, which are used in wound healing. So if you're very young, if you're a toddler or a very young kid, and you cut yourself and have a bad gash on your hand or your arm or whatever, your mobilization of your innate stem cells for wound healing, the mobilization of the stem cells in your body which are required to make fibroblasts and then carry out the wound healing process, occurs very efficiently. Yeah, so you tend not to scar, the wounds tend to heal very well, etc. As you progress through adulthood and get into old age, the mobilization of innate stem cells tends to decrease somewhat, so it's just not as efficient a process, which means that if you cut yourself, you may end up with a scar, whereas the same kind of injury in a younger person might not have resulted in such a visible blemish on the skin. So all down to the efficiency by which these innate stem cells are mobilized. One idea, therefore, is the possibility of increasing the mobilization of innate stem cells to make the process more effective. So in other words, you could boost wound healing by making the mobilization of innate fibroblast stem cells more efficient, or maybe even mobilize innate stem cells and then modify them so that they're able to become other tissue types other than fibroblasts. So this is like an area that's not been explored really clinically as much as the simple replacement technology we talked about, the example above here. But this idea has obviously got great potential benefits as well. Not least, you could try and create your own Wolverine if you were of that particular inclination. It's also obviously got, if you're a cynic, connotations for the military. If you're able to boost wound healing in military personnel or during surgery, for example, or in people who are suffering from haemophiliacs, and other situations where improved wound healing would be beneficial to an individual. So it's just to give you an idea that there is more than one way to skin a cat sort of thing. It's another angle on which cell-based therapy has got potential benefits in the future. So nuclear reprogramming is very important then with regards to our modern appreciation of cell-based therapy. So I want to talk a little bit about what nuclear reprogramming is and how we first really got turned on with regards to its potential benefits. It's essentially the concept that any cell can be reprogrammed to return to a pluripotent state. So a more simplistic way of saying that is that cell fate isn't set in stone. So there was a time that scientists believed that once cells differentiated into a specific cell type, that was it. So once a heart cell became a heart cell, it was a heart cell forever. Once a neuron became a neuron, it was a neuron forever, etc. The process was unidirectional. It didn't go both ways. We now realise that that's not true. The first real big finding, or one of the big scientific experiments which really brought this to the public's, brought it to the wider attention of the general public, was the cloning of Dolly the sheep in the late 90s. So what Dolly did, really more than anything else, and certainly from your point of view with regards to this module, what you need to realise is that Dolly proved that nuclear fate is reversible. So what Dolly the sheep proved was that if you have a nucleus in a cell, so let's say you have a nucleus in a heart cell, you should realise by now that whilst our genomes are identical in every single cell in our body, it's the transcriptional program, the genes that are being expressed and repressed in individual cells differ. It's called the transcriptome. Yeah. So a heart cell will be expressing and repressing different genes than a liver cell and a neuron, etc. That's what makes a heart cell a heart cell. Yeah. So what scientists did is they took a nucleus from an udder cell, from a sheep, Dolly's mum, inverted commas if you like. So this is a nuclear material, the nucleus from an udder cell. So this nucleus will be producing all the genes, proteins, etc. required to make an udder cell, make it work. And they put this nucleus, this udder cell nucleus, into an enucleated egg cell, so an ovum. Yeah. And what they saw was something quite remarkable. They saw that the nuclear material from the udder cell kind of got reprogrammed in such a way that it then caused the development and division of the egg cell, and eventually the egg cell went on to become Dolly the sheep. Yeah. So for a long time, scientists would not have expected that to happen. They would have thought that the nucleus from the udder cell could only act as an udder cell nucleus, and it wouldn't be able to undergo the complex, very complex changes required to its transcriptome, to the genes it's expressing, etc., to its epigenetic modifications, a methylation pattern in that genome required so that it could then act to cause the division of this egg cell into a new sheep. But nevertheless, that is exactly what happened. This udder cell nucleus caused the egg cell to become to act as an egg cell and to make a new sheep. There will be there should be a picture of Dolly the sheep here in the actual lecture. So for part-time students who won't see that lecture, I apologise. Just imagine there's a sheep here. Dolly the sheep looks exactly like every other sheep. You know, I can't tell the difference. I'm hoping you can't either. I've just forgot to put this on the podcasted version of these slides. So whilst Dolly was like any other sheep, she was different in some ways. Some people would argue with regards to her health, and this is one of the things that people like to discuss with regards to putting the brakes on this sort of technology, because we don't fully understand the implications for health and how it's going to affect the individual. So Dolly actually died in 2003, so she was only 6 years old, of a virus-associated lung cancer, but she was kind of plagued with health issues before that. Some argue that these health issues were a consequence of the environment, so she was fat, for example. People say she's fat because people used to give her treats all the time because she was a bit of a celebrity. But she also developed early onset arthritis, which is a little bit more difficult to explain. And perhaps most strikingly of all, when people actually looked at Dolly's DNA, and in particular the telomeric regions of the chromosomes, so remember telomeres are the ends of chromosomes, and it's the shortening of these telomere sequences which is associated with the aging of the cell. So when they looked at the telomeric regions of Dolly's chromosomes, they saw that the telomeres were far shorter than you would expect in a sheep of Dolly's age. And what people postulated then was that perhaps Dolly was actually older than we thought, even though she was only 6 in natural years. Perhaps her genetic age was a combination of her actual age and the age of her mother when the DNA was extracted from that other cell initially. No way of proving that, but that was one possible theory. Certainly she had health issues, and eventually she died of an unrelated, potentially, illness, but not before giving everybody food for thought regards to cell fate, etc. So it's now this guy really that I want to focus on. So this is Shinya Yamanaka, and what Shinya Yamanaka did in 2006 is really turn all of this nuclear reprogramming, cell fate, cell-based therapy field on its head. Because what Professor Yamanaka showed was that not only can nuclei be reprogrammed, but we can actually reprogram cells themselves. Yeah. So he showed that reprogramming of cell fate was possible, and he did this by basically taking a mouse fibroblast cell and expressing several genes in that cell, which caused the mouse fibroblast to change from a basic fibroblast back into a pluripotent stem cell. So this really got a lot of people very excited for obvious reasons, because now we had a way potentially of making pluripotent stem cells that didn't require destroying embryos. The other thing that really got people excited was how simple the thing was. So all Shinya Yamanaka did was express 4 transcription factors. And by expressing just 4 transcription factors, he managed to convert a very simple fibroblast cell into a really, potent, pluripotent stem cell, with limitless potential in regards to the different cell types it can become. So without going into the actual specific details, you can imagine that there's a really humongous amount of genetic reprogramming going on to make that happen, to change a fibroblast into a pluripotent stem cell. It's a completely different transcriptional program, completely different methylation pattern for the genome, and yet all of these changes can be achieved by just these 4 transcription factors. So Shinya Yamanaka's work's been repeated using human fibroblasts now, so we can now make human induced pluripotent stem cells, or iPS Cs, using a very similar technique by expressing the same 4 basic transcription factors. So when you see this term, like you see in the lecture title, iPSC, it stands for induced pluripotent stem cells. And these are pluripotent stem cells not derived from the embryo, but induced from other cell types, fibroblasts, although others exist as we'll talk about, via the expression of 4 transcription factors. At least in the first instance, 4 transcription factors. So, moving forward a little bit since 2006, it's a relatively new area anyway, obviously. I guess you guys would have been, just about to embark on your GCSEs when Yamanaka was publishing this work in 2006. We've come a long way in the last 5 years. It shows you how quick science moves these days, especially when there's a lot of potential biomedical or medical benefit, pharmaceutical companies get interested, etc. We now understand a lot more about how the actual process works with regards to the reprogramming of fibroblasts to induce pluripotent stem cells. It requires, in essence, mesenchymal to epithelial transition between the cell types. People have managed to tie down a little bit what these transcription factors actually do to enable this transformation from fibroblast to stem cell as well. So these are the 4 transcription factors that Yamanaka used in his initial experiment on mice: SOX2, OCT4, MYC and KLF4. Other transcription factors have been used now as well. Fewer versions of these 4 have been used in certain cell types. I'll mention that a little bit later on. But if you like, this is the pure experiment. So if you're talking about the canonical, the basic experiment for nuclear reprogramming, you will be talking about the expression of 4 transcription factors: SOX2, OCT4, MYC and KLF4 in the fibroblasts, and the expression of these 4 eventually cause these fibroblasts to turn into pluripotent stem cells. We now know that these 3 transcription factors, SOX2, OCT4 and MYC, work by inhibiting this TGF-beta signaling molecule here, and that basically, in conjunction with the activation of the E-cadherin via the KLF4 transcription factor, enables this transition from mesenchymal to epithelial, which is important in the overall transformation of fibroblasts into stem cells. One thing that is important to note is that each of these transcription factors can also function potentially as oncogenes. So that's obviously a major concern with regards to this technology, and we'll talk more about that later on. In particular, MYC, you should have heard of. KLF4 is also a potent proto-oncogene. So it's not, as always with these things, it's very exciting that we have these 4 factors that can allow us to generate these pluripotent stem cells, but it does come at a cost. Yeah, so we'll talk more about how we get around that cost later on. Later on, being now actually. So nuclear reprogramming, what challenges do we face currently? Well, the first thing to think about when we talk about nuclear reprogramming then is what's the best cell to reprogram. So Yamanaka originally did his work on fibroblast cells, but are they really the best ones to reprogram? Would other cells provide a better option? So for example, people have looked at reprogramming blood cells. So blood cells are convenient because they're very accessible. It's very easy to get hold of people's blood. Yeah, so that would be a big advantage if we could just take blood cells and reprogram them to generate induced pluripotent stem cells. And several studies have managed to do that. Take blood cells, lymphocytes, and reprogram them into pluripotent stem cells. Similarly, another really useful source for reprogramming pluripotent stem cells would be any cells really associated with the skin, because again they're very accessible. Keratinocytes have proven to be very good at being reprogrammed into induced pluripotent stem cells. The process is very rapid, a lot more rapid actually than for fibroblasts. So there are a lot of potential benefits to the use of keratinocytes in this technology rather than the fibroblasts which were used in the original study. Finally, neural stem cells as well. So we can take a tissue stem cell, something that's not got as much plasticity as a pluripotent stem cell, and we can reprogram that to send it even further back, or turn the developmental clock of that cell even further back, so that it goes from being a neural stem cell, a tissue stem cell, back to being a pluripotent stem cell. So the big advantage of using neural stem cells to generate pluripotent stem cells is that we don't now require 4 transcription factors in order to cause that transformation to occur. We only require 1 transcription factor in order to have that transformation occur. So less transcription factors to express in the cells, less issues around expression of those transcription factors, less potential problems with regards to the fact that many of these transcription factors act as proto-oncogenes. So it's not just fibroblasts that can be transformed back into induced pluripotent stem cells, other cell types can as well. So we need to think carefully before we start treatment which cell type we should be working with, and this is something that anybody who's running a clinical trial would want to address. If you like, one of the major obstacles with regards to nuclear reprogramming is actual delivery of the reprogramming, inverted commas, genes themselves. So delivery of OX4, SOX2, MYC, KLF, etc. How do you get these cells into a fibroblast cell or a blood cell or a keratinocyte in the first place in such a way that they then become expressed and enable reprogramming of that cell type? Well, initially at least, Yamanaka and other people working in the field used the retroviruses to deliver these different reprogramming genes to their fibroblast cells. So as we talked about in the gene therapy lecture, viruses, in particular retroviruses and lentiviruses, represent the major gene delivery vehicle still for anybody who's interested in trying to deliver and express genes in human cells or animal cells. However, they all come at a cost. So we talked already about how retrovirus integration can lead to problems. Obviously, overexpression of a proto-oncogene is one of the ways in which you can activate a proto-oncogene, make it become oncogenic, and then cause transformation of the cells. Retroviruses will express these reprogramming genes at very high levels and will continue to express these reprogramming genes at very high levels once the cell type itself has been converted from a fibroblast into a stem cell. So how do we get around this problem? Well, there are other options that we can use that are different from retroviruses, and it's only really in the last 12 months, 18 months that publications in Science and Cell, so the real big hitting journals, have started to emerge, whereby groups have shown that they're able to create these induced pluripotent stem cells via expression of these reprogramming genes in a method that differs to retrovirus transduction, so introduction via a virus. One of the ways in which these 4 genes have been successfully introduced is via electroporation. The advantages of using electroporation are that you don't have any virus at all present there. There's no opportunity for the virus to integrate. You've no virus to clear after the transformation events occurred. So after the DNA has been introduced, the transfection events occurred. The downside of it is it's nowhere near as efficient. Electroporation is very inefficient. Certainly you wouldn't be able to use this method for the mass production of stem cells, which, as we'll discuss in a slide or two's time, is an important thing to consider as well in this field. What other options then exist? Well, there was a really interesting paper in Cell in the last year or so, whereby scientists made use of a particular technology called Crelox recombination to be able to deliver initially the 4 reprogramming genes required via a retrovirus, using a retrovirus, but then once their fibroblast cell had been converted into a pluripotent stem cell, they could then get rid of the integrated retrovirus completely to effectively clear both the viral genome and the 4 reprogramming genes from the pluripotent stem cells. So remember, these expression of these reprogramming genes is required to drive the changes necessary that convert a fibroblast into a pluripotent stem cell. Once you've got a pluripotent stem cell, you don't need to continue to express these things. In fact, continuing expression of these 4 things is probably going to be a bad thing in the long run, because it's going to make it's something artificial in that stem cell that wouldn't be there normally, and may interfere with potential treatment or other uses of the technology. So I'm not sure if you've encountered Crelox before, probably not. I don't want to go into any detail other than what appears in this diagram here. Effectively, you can tag either end of a piece of genetic infant sequence, and that could be a retrovirus genome which contains the 4 reprogramming genes we want to express. You can tag either side of that retrovirus genome which contains those reprogramming genes with these Cre elements, these little orange arrows here on the diagram. And then you can use a special type of recombinase enzyme called Cre recombinase, and by adding the Cre recombinase you can cause these 2 Cre elements here to recombine and excise out whatever lies between them. So in this case it's exon 2 in this example. Imagine exon 2 is actually the retrovirus genome which contains the reprogramming genes. Once you've got the pluripotent stem cell converted from the fibroblast, add Cre recombinase. That will cause recombination of these 2 elements, and it will chop out the retrovirus genome, leaving you with just a nice intact pluripotent stem cell genome without any trace of retrovirus sequence or the OX4, SOX2 reprogramming genes either. So you go back to, if you like, just a stem cell as it would appear if it was an embryonic stem cell. And in the Cell paper, which I'll put on the extra reading material, these guys show that these stem cells, the stem cells that they generate using this Cre recombinase technology, actually resemble genuine embryonic stem cells to a higher degree than those induced pluripotent stem cells, which are still expressing these reprogramming genes and contain retrovirus sequence. So they're a more accurate representation of what an actual induced pluripotent stem cell looks like. So that's a nice example of how they've got around that problem. So what's the current situation then with this technology? Well, the bare bones of the fact are, and some of you may realise this already, that stem cell therapy or cell-based therapy has been around for donkey's years already, in the form of bone marrow transplantation. So if you're unfortunate enough to have chemotherapy or radiotherapy at any point in your life to treat cancer, one of the consequences of that can be that your hemopoietic stem cells, the stem cells in your bone marrow, get wiped out. So remember, chemo drugs, radiotherapy, all works on the premise that cancer cells divide very rapidly, so let's use drugs which kill rapidly dividing cells. Rapidly dividing cells include cancer. It also includes hair follicles, that's why your hair falls out when you have chemo. Also includes hemopoietic stem cells. So the stem cells in the bone marrow are constantly replacing your erythrocytes in your blood at a phenomenal rate. Billions upon billions of erythrocytes are replaced every few seconds by hemopoietic stem cells. It's a mind-bogglingly high rate of turnover. So obviously when you blast a patient with chemicals or radiotherapy that effectively destroy rapidly growing cells, one of the cell types that really get wiped out are these hemopoietic stem cells. A way around that is to give a bone marrow transplantation from a relative who's a bone marrow match. So you blow out the hemopoietic stem cells and hopefully the cancer, and after the treatment you give the person bone marrow back, the hemopoietic stem cells come back and they can continue functioning as normal. That's basic cell-based therapy, been around for years. There's been some reported successes historically as well of taking this therapy a little further. Some people thought that perhaps that you'd be able to use these hemopoietic stem cells, these bone marrow cells, to treat heart attack patients. So one of the things that happens when you have a heart attack is your cardiomyocytes basically become damaged, they die, your heart doesn't work as well anymore. Heart tissue doesn't divide, so like neurons, your heart cells, your cardiomyocytes never undergo mitotic division. So one of the reasons why you don't get cancer of the heart, because effectively your heart never divides, so there's never opportunity for genetic damage to be inherited and for tumours to appear. The downside of that is there's no way of regenerating itself. If it has an attack, these cardiomyocytes remain damaged forever. So some people came up with the idea of maybe using bone marrow stem cells to try and regenerate cardiomyocytes, and some groups claim that they saw positive results with this, but it was controversial and not really conclusive. But it's really now the development of these induced pluripotent stem cells, Yamanaka's work, so his ability to generate, in theory, lots of pluripotent stem cells, human pluripotent stem cells, in an ethical way that doesn't impinge or require the use of embryos, that have provided three big major opportunities for biomedical science research. So these opportunities are better disease modelling, better drug screening for diseases, and regenerative medicine. And these are kind of summarised in this cartoon here. So we've got this idea then that you can have a patient from which you can isolate skin fibroblasts very easily. You express SOX2, OX4, KLF4, MYC, etc. in these cells. That causes your fibroblasts to become these pluripotent stem cells, and then these pluripotent stem cells can literally become any other cell type of interest. They can become fibroblasts again, they can become blood cells, they can become oligodendrocytes, they can become neurons, they can become myocytes, cardiomyocytes. They can become anything you're interested in. Once you've got a specific cell type, you can use that specific cell type to do studies on disease cells, as we'll talk about in a second. You can use it for cell-based therapy, give it back to the patient so that you get a working copy of a cardiomyocyte to replace the ones damaged after a heart attack. Or you can use it for doing drug screening potentially as well. So I'm just going to discuss these three different uses now at the end of the lecture for the next 5 or 10 minutes. So to start with then, disease modelling. Well, the first thing to realise is that we have mouse models for modelling human disease. There are issues around the ethics of using animal models anyway. Some people are really against this. And certainly not all human diseases can be accurately modelled in animals. It's true that you know not every disease can. Parkinson's disease, for example, there is a mouse model for it, but it's not particularly informative for the disease. So the beauty then of these induced pluripotent stem cells is that, in theory at least, we can generate an induced pluripotent stem cell from a patient who's afflicted with a disease. So, for example, Parkinson's disease, yeah, and then we can isolate fibroblasts from a Parkinson's patient, generate induced pluripotent stem cells, and then differentiate those pluripotent stem cells into a neuronal cell, yeah, so the cell type which is affected by Parkinson's. Now remember, for a genetic disease, yeah, so if it's a disease which is caused by a sole genetic defect, that genetic defect is going to be present in every single genome in the cell type, 99% of the time, for the vast majority of cases. So if you take a fibroblast, the genetic mutation will be in the fibroblast. Yeah, it might not be a problem for the fibroblast because the particular gene which is affected might not function in that cell type. For example, CFTR, cystic fibrosis, it's not going to be an issue for a fibroblast if that protein isn't present because it's not been expressed in that cell type. Whereas it is a problem for a lung epithelial cell or a pancreatic cell or the vas deferens cells. Yeah. But the point is, you can take a fibroblast cell from a patient who's got a genetic disease, generate pluripotent stem cells, and then differentiate the stem cell into the cell type that is affected by that disease. And what you then generate is, at will, almost unlimited reservoirs of the tissue type that's affected by the disease for you to actually model and do disease studies on. So I mentioned Parkinson's earlier. It's not the best example because one of the things where this still doesn't work very well is for complex diseases. So complex diseases are still tricky to model. And the reason being that it's not just genetic factors which have given rise to the manifestation of that disease, is the environmental contribution as well. And that's nicely exemplified for Parkinson's, where people have taken fibroblasts from patients who have got late stage Parkinson's disease. They've generated induced pluripotent stem cells from these individuals, and then they've differentiated these stem cells into neurons, and they've actually shown that these neurons are dopaminergic, so they resemble Parkinson's neurons, but they actually lack the functional changes which are present in the neurons of Parkinson's patients. So in other words, the neurons that you generate from stem cells from a Parkinson's patient look the dopaminergic. They look like Parkinson's neurons, but they don't have all the functional changes which a Parkinson's neuron has when you just take it from the brain of a cadaver who died of Parkinson's disease, or isolate tissue from people who are suffering from the illness. The reason for this is that these cells haven't been exposed to the environmental factors that the neurons of a Parkinson's patient has. So one of the ways around this might be to generate a dopaminergic neuron from a Parkinson's patient and then expose it to reactive oxygen species, some of the chemicals from cigarette smoke, from pesticides, the sorts of environmental things which are known to also trigger and lead to the disease. Yeah, so those are the sorts of things you need to consider as well when looking at disease modelling using induced pluripotent stem cells. However, there are benefits as well to this. So other benefits, developing disease models as well may allow researchers to actually model pre-symptomatic abnormalities. So for example, Huntington's disease might be a good example of this. If you have a patient who's 20 years old, they get a positive test for Huntington's of 3940 CAG repeats in the HTT gene, that patient is going to get Huntington's disease. However, it's not going to manifest until they're probably around 39, 35, 40 years old, maybe a little older if they're lucky. The point is, if we can isolate neurons from a 20-year-old with the genetic changes, which we know are going to eventually lead to Huntington's disease in later life, we can do experiments on those neurons. We can look for very early changes in the neurons, biomarkers which tell us when the changes are happening, and try and get a better idea of how the disease progresses very early stage. And if we can do that, if we can get a better understanding of the mechanisms which behind early onset of Huntington's disease or other similar diseases, we might be able to get better diagnostic tools for monitoring when patients are starting to develop symptoms, and also develop better drugs which can treat and manage the very early symptoms of those diseases, maybe even extend life or treat the disease in the long run as well. So we just wouldn't be able to do that normally. You can't go around chopping people, taking a 20-year-old who's been diagnosed with Huntington's and isolating brain tissue from that person and doing experiments on it in a tissue culture flask. You just can't do that. But with induced pluripotent stem cells, we can do that, because we can get a fibroblast from that person, from the skin. We can convert it into a stem cell and then change it into a neuron, and that neuron is that person's neuron, with those genetic changes to the Huntington's gene, and then we can do experiments in the lab to try and monitor and work out what's going on with the disease. Really powerful, exciting stuff. Drug screening then. So another real big benefit to screening of drugs. Some things to consider though with regards to drug screening, and this goes hand in hand with the disease modelling slides, comments about disease modelling on the last slide. It's not always going to be possible to recreate disease phenotype in vitro. So just because we're able to isolate fibroblasts, make them into neurons and convert them back into neurons, blah blah blah, and say those neurons come from a patient with a neurological disease, it's not always going to be the case that those neurons grown in tissue culture are going to mimic or give us a good representation of what that disease is like in vivo. In other words, how accurate are any kind of drug studies going to be on the in vitro cell line compared to what the effects of that drug would be in a person? The second thing to consider with drug screening, pharmaceuticals don't just have 3 or 4 flasks in an incubator and test 5 or 6 drugs on those flasks every other week. They want to test tens of thousands of drugs on tens of thousands of individual cell lines in a matter of hours using robotics. So high throughput screening is essential for modern drug screening programs. Can the rate of iPS production match this rate of screening that's required in the pharmaceuticals? Are we actually physically able to produce these induced pluripotent stem cells at a rate that can match the needs of the pharmaceutical for Freudian slip industry? That's something we need to consider. And also you've got to realise that the purity of these pluripotent stem cell samples isn't 100%. So if you isolate fibroblasts and convert them back to pluripotent stem cells, what you actually get is a heterogeneous culture of cells. Some of them have gone all the way back to pluripotency, some of them are stuck earlier on or later on, whichever way around you look at it, in the developmental cycle. Yeah. So you don't either have 100% pure fibroblasts in one flask, express those 4 genes, and then get 100% pure pluripotent stem cells in the other flask. You have a heterogeneous mix, and the fact that the sample's not pure could lead to false positive results in regards to the drug screening. You might see an effect of the drug that's due to the fact it was acting on an immature pluripotent stem cell rather than the real thing. This is a nice figure here. It nicely summarises the benefits of drug screening and cell-based therapy, which I'll talk about in the next slide, with regards to pluripotent stem cells. For drug-based therapy here, you can see the idea is you would isolate cells from the patient and generate patient-specific pluripotent stem cells. These can be differentiated into the specialised cells of interest. So say this person had Parkinson's, we differentiate them into neurons. Those neurons could be put into a drug screening program then to try and see which drugs are effective against these diseased neurons, effectively reproducing the disease phenotype, and trying to see which drugs work best. And if you get a drug which works best against the Parkinson's neurons, then there's a potential for that drug to actually treat all patients with Parkinson's, not just the patient who's initially had the disease here. Tissue regeneration is slightly different. We'll talk about how this figure here fits in with that in a second. There are some risks associated then with the concept that we can just reintroduce pluripotent stem cells into patients to replace damaged tissue. Mentioned earlier on, teratomas, those disgusting looking tumours at the beginning with teeth and all sorts, lips and all sorts. Looks like a kebab from your local kebab shop. These teratomas, they're a major risk really. They're one of the major risks for tissue regeneration studies using these induced pluripotent stem cells. Studies in mice have shown that when you actually introduce these induced pluripotent stem cells into immunosuppressed mice, tumours form, teratomas form. And there's a real concern that the same thing's going to happen in humans. There aren't really, as we'll see, enough clinical trials yet to see whether that's going to be the case or not. But it's quite possible that it will be the case. There's also this concept, obviously one of the strong concepts behind induced pluripotent stem cell technology is that you're actually using the patient's pluripotent stem cells to generate new tissue to potentially replace an organ. So this is different to organ transplantation. You know that you have to have organs donor matched, HLA matched, so that the organs aren't rejected. One of the golden, gooses about, or the great ideals of stem cell based therapy is that the stem cells are specific to the individual, so there will be no immune response because these cells are the patient's cells. But can we really be sure of that? If we've made these changes to the fibroblasts using these 4 transcription factors, if there's residual virus, genomic information left over, could that not subtly change the immunogenic profile of these cells so that actually they're not completely non-immunogenic? So we don't know yet whether or not there might be an issue with rejection for patient-specific induced pluripotent stem cells. There's not enough information out there, but we need to be aware that that might be a problem. I mentioned earlier the problems around integrating differentiated cells into solid tissue organs. The fact that the heart, the brain, the pancreas are complex organs, and might not just be the case that these healthy individual cells are going to integrate properly in those tissues. And in fact, people have started doing some really exciting research now using tissue scaffolds. So rather than relying on these cells just integrating by themselves, we can grow tissue on a tissue scaffold. So people have used scaffolds to grow artificial lungs now, and pancreases and all sorts of things which originally are derived from individual differentiated cells that form around a scaffold to eventually differentiate into a mature working organ such as a lung. So this might be the way that we go around it, rather than introducing these cells into individuals in an individual cell basis. We use the cells in a bioincubator to grow the organ, then the organ gets put in. Yeah, all sorts of things to consider. So just to finish with, I want to give you a couple of examples of some of the current studies which are underway for induced pluripotent stem cells, some of the ways in which it's been used. This table on your right, you don't need to memorise all the information in this table. It's from an article which I've used heavily when I've written this lecture. There's not a lot of information on this stuff in core textbooks. It is mentioned in the human molecular genetics core textbook by Strachan and Reed. There's a nice section in there. But it's such a new area, you need to really look at some additional reading as well if you want to get your head around this topic. This table and a lot of the other lecture materials taken from a really nice review that I'll put on the extra reading list. I think you should all have a look at. That's very interesting. And this lists some of the diseases that have been modelled now using induced pluripotent stem cells. These are published examples, and you can see there's a whole range now of diseases which have got induced pluripotent stem cell models up and running. There are very, very few examples of pluripotent stem cells being used to treat actual diseased cells, so if you like, for tissue regeneration. 3 examples which are currently or have just recently finished or just currently underway include from a company called Geron. We've been trying to use oligodendrocytes to repair spinal cord injuries. These guys run into a lot of trouble to get FDA approval for the trial after they showed that the introduction of these oligodendrocytes into mice led to the formation of microcysts at the site of introduction. So the FDA made them jump through loads of hoops before they could put this trial into humans, and they're still involved in those negotiations with the FDA. A company called Advanced Cell Technologies are trying to treat age-related macular regeneration, which is a real big disease with regards to blindness in later life. And these guys are using pluripotent stem cell derived retinal epithelia to treat this disease. And finally, there's another trial which is just about to go underway to use induced pluripotent stem cells derived from the pancreas, or pancreatic pregenerator cells, to treat type 1 diabetes. So these are the 3 examples of actual clinical trials that are underway at the moment. So just to summarise then, so the aim of cell therapy is to replace non-functional or dead cells with healthy cells and thereby cure disease. Does this by using stem cells, and in particular these pluripotent stem cells, which have got the potential to turn into any other cell type. Now, for many years, the use of pluripotent stem cells was really limited due to ethical reasons. You had to get these things from embryos. But the development of induced pluripotent stem cells, the work of Yamanaka, and the expression of these 4 different transcription factors in fibroblasts has really turned the entire field on its head in these last few years. So there's loads and loads and loads and loads of money and research being pumped into this field now. Certainly if you're interested in doing a PhD, this sort of area is something that's very exciting and would be a good topic to perhaps consider working on. There are a couple of clinical trials going on at the moment using these induced pluripotent stem cells, and these were approved in the last 12 months, so it gives you an indication of how current this stuff is. Okay, so thank you for your attention. I hope the audio is not too unclear on the podcast, and enjoy reading the extra material that will be placed on Backboard for these lectures. Thank you.